Welcome children. In this video, I will be dealing with skeletal muscle, the structure and the proteins which constitute the skeletal muscle. So, let us get started. This is the structure of the skeletal muscle. I am zooming through it. You can see this is the skeletal muscle which is attached to the bones. Each skeletal muscle comprises of muscle bundle cells, and each muscle bundle cell consists of muscle fibers. So, you can see like a hierarchy. This is the skeletal muscle, and I am going to pull out one skeletal muscle bundle cell. And each muscle bundle cell comprises the muscle fibers. And this muscle fibers appears to be striated because it comprises two sets of myofibrils. This is the thick, which I am shading with a dark pink color, and the thin, which I am shading with light pink color. The thin filaments are called actin filaments, and the thick filaments are called myosin filaments. These filaments are arranged parallelly, alternatively, side by side, with the edges overlapping. So, once again you can see the skeletal muscle, and the same skeletal muscle comprises a number of muscle bundle cells. So, I pulled out one muscle bundle cell, and each muscle bundle cell comprises a number of muscle fibers. I pulled out one muscle fiber, and in each muscle fiber, which is the smallest unit, structural and functional unit of the skeletal muscle, comprises of thick and thin filaments called myofibrils. They are myosin, which are the thick filaments, and actin, which are the thin filaments. How are they arranged? They are arranged parallelly, alternatively, side by side, with their edges overlapping. So, this is the picture which I pulled out. This is the entire muscle in cross section. Then a bundle cell is pulled out, and from the bundle cell we have pulled out a muscle fiber, and on the muscle fiber I arrange the two sets of myofibrils. As I have informed you on, the two sets of myofibrils are thin and thin filaments, that is the actin and myosin. So, I will roughly draw that striated appearance or the ultrastructure of the muscle fiber. Continuing with the same coloration, pink, I am going to indicate the thick filaments. 1, 2, 3, 4. 4 thick filaments. Adjacent to it, the thin filaments. 2, 3, 4. Here also 1, 2, 3, 4. Now, I am going to label all the parts in this striated ultrastructure of the muscle fiber. Now, you can focus at this region. This region, correctly to say only the region which comprises, I am doing the adjustment to show you only the region which comprises of the thin filaments. See, I have indicated with a rectangle. This region is called the I band. And this region, I'll draw it here. I'm widening it to accommodate the edges. This is called the A band. I hope you understood the I band and A band. I band exclusively comprises of only the thin filaments. See, thin. Only the thin middle portion, not the overlap region. Overlapping is continuing here, 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 here. Only I highlighted only the central region which comprises only of the thin region or thin filaments, that is called the I band. And this side, I have focused on the A band. A band a better picture of the A band is this, where I focused on the thick filaments with the edges overlapping the thin filaments. See, this is the correct A band, and A band stands for anisotropic band. See, it has, if I am indicating with the same pink color, it's consisting of the full length of thick filaments. See, full length of thick filaments, and also the edges of the thin filament. So it is called anisotropic. So the name is justified, anisotropic. Like we say heterotropic, this is anisotropic because it comprises full length of thick filaments and overlapping thin filaments. Whereas this is isotropic, like homogeneous. It comprises of only the thin filaments. See, only the thin. It indicates only the thin. The other part of the thin which is overlapping with the thick is outside. This is hence called the isotropic band. So I denoted the I band and A band. Now in the center of the I band is a line called the Z line. Correctly at the center there is a Z line. So at the center of every I band there is a Z line. And the portion of the muscle fiber between two Z line is called sarcomere. Very, very important. It is the contractile unit or structural unit of contraction. So again, this is the second sarcomere. So the distance between consecutive Z lines is called the sarcomere. So depicted on the board are 3 sarcomeres. So sarcomere is the portion between two Z lines. So that's all about the I band. Now moving on to the A band. Please focus at the A band. You can see in the A band, it consists of the entire length of the thick filaments or myosin filaments and also the overlapped thin filaments. So in this particular band, A band called anisotropic band, the central region which consists of exclusively thick filaments is known as the H zone. So within the A band, the central part which consists of only the thick filaments is called the H zone. The sides there is overlapping of thin and thick. So I am not talking about the thin and thick side regions, only the central region which comprises the thick filaments is called the H zone. And just like the I band, at the center there is a line called the M line. So center of every A band there is the M line. I hope you have understood the striations or the ultrastructure of a muscle fiber. Once again, there is alternate dark and light striations. The dark band is the A band and the light bands are the I band, and if you focus, the I band exclusively comprises of thin filaments or actin filaments, and the central line that is running through it is called the Z line. Talking about the A band comprises of that region that has the full length of the myosin or thick filaments and at the edges overlapped with the thin filaments. This entire banding is known as the A band, and within the A band the central region is slightly not so darker than the edges because it is exclusively of thick filaments. That is called the head zone. So, head zone is found in the middle of the A band, and right at the center of the A band is the M line. So, the same is indicated in this pull out. I am zooming through the pull out. The very same thing, there is the very thin filaments which constitute the I band, the very thick filaments which constitutes the A band, then the Z line which runs through the thin filaments. See the Z lines, you can identify the Z line because it runs through the thin filaments and they demarcate the sarcomeres, and there is M line which runs through the thick filaments. So, remember the lines M line running through the thick filaments. So, here again I am clearly indicating the line running through the thick filament is the M line, and the line running through the thin filament is the Z line. And Z line is very remarkable or significant because the region between two adjacent Z lines is known as sarcomere, which is the structural unit of contraction. Now, the next slide we will be dealing with the structure of actin and myosin filaments which constitute the muscle fibers. They are also called myofilaments or myofibrils. So, first let us deal with actin filament, which is commonly called thin filament. It comprises 4 proteins, G actin which stands for globular actin, F actin which stands for filamentous actin, tropomyosin and troponin. So, once again the 4 proteins that constitute the polymer actin or thin filament are globular protein, filamentous protein, tropomyosin and troponin. Now, I am going to construct an actin filament with some aids. Let this be the globular protein. So, this globular bead-like structure is the G actin. It is the monomer of the F actin. So, many such units repeated will form the F actin. So, from the very name you understand this globular protein is the G actin, and many such repeated units form the F actin called the filamentous actin. So, this will be constructed into this structure. Now, there are 2 such F actins which coil around each other helically. So, the structure that I am going to indicate next is this. 2 F actins coiled around each other to form the F actin. So, from G actin I built the F actins, 2 of them helically coiled. So, going into the construction. See, this is the one F actin formed by the monomeric units of G actin. Many such a number of G actins together form this peptide or this chain of F actin. That way, there are 2 such F actins or filamentous actins, and these are not running parallelly. It is not running parallelly, it is running helically. We are going to coil it. So, now these 2 are F actins which are parallel. I have to helically wind it. So, you can see I am winding it helically around each other. Now there is a third protein, that is the tropomyosin. See, the ribbon which I have indicated is tropomyosin, and you can see here and there tropomyosin I have put one ball. These balls are called troponin. So once again this blue ribbon is tropomyosin, and on this blue ribbon which indicates the tropomyosin, there are active sites, and on these active sites you can see one ball. What is this? Troponin, complex proteins. Like that there are two such tropomyosin. This is one tropomyosin and this is the second tropomyosin, and you can see at intervals there are active sites, and on these active sites we can see these troponins bound. I hope you have understood. Now this is also going to be constructed. This is also going to run very close to the helically coiled F actin. See the way all are packed together. See, this forms a complex polymer called actin. I hope you have understood actin. There are 4 proteins constituting actin. So we have now constructed an actin, a complex protein or filament which comprises of 4 proteins. The 4 proteins are G actin, which got organized into F actin. Like that way there were 2 F actins which got helically coiled to form 2 filamentous actins, and then around them, running across or wrapping across them, is the blue color structure. What is that called? Tropomyosin. And tropomyosin at intervals are attached to complex proteins. See, here and there complex proteins which are indicated with thermocol balls, they are called the troponin, and these troponin are bound to the active sites. So they are tightly bound. I tried to remove, it's tightly bound to the active sites found on the tropomyosin. So with this you got a clarity on the structure of actin filaments. Now moving on to myosin filaments. Myosin comprises of monomeric units called myromyosin. It comprises of a number of monomeric units called myromyosin. And myromyosin comprises of a globular head, a short arm and a tail. This is the structure of one myromyosin. So many such myromyosins are organized together. So this is the tail, short arm, globular head. Tail, short arm, globular head. Tail, short arm, globular head. And I'll join these together to form the polymer called myosin. So now I got a myosin filament. So the structure of myosin is very simple because it is a polymer of myromyosin, and one myromyosin comprises of 3 parts. The 3 parts are the globular head, short arm and the tail. Of these, the globular head and short arms form the heavy myromyosin and the tail forms the light myromyosin. So I have a model of one myromyosin. So this is the globular head, short arm and tail. So this is one unit or one monomeric unit of myosin called myromyosin, and this constitutes the heavy myromyosin, the globular head and the short arm, and the tail constitutes the light myromyosin. Like that, if I make a number of repetitive units, I get many of such structures. Okay, so this is the way the myromyosins are organized to form the myosin filament. And here importantly, the function of the globular head, it is a site for ATP to bind, and also it is site for actin. So the role of this globular head is very, very important. Globular head is also known as ATPase. It serves as site for ATP and also site for actin. Now with the pullouts, I will show you the structure of both thin filament, which is the actin, and thick filament myosin. So I told you we started off with the globular proteins. See number of globular proteins like the beads I showed you, and they got organized to form the F-actin. Two F-actin chains were coiled around each other, helically coiled, to form the F-actin. Now we have the troponin, tropomyosin. See the tropomyosin, which is indicated with another color called green color, which is attached here and there at intervals with troponin. So this blue is indicating troponin. And finally we have the structure, organized structure. The final structure, which comprises of the globular protein called G-actin, which got polymerized to form one F-actin, like it two F-actins got helically coiled. See the way they are coiled, helically coiled. And close to this, there is another protein called tropomyosin. See, that is also helically coiled, very close, in close association. That is also 2 in numbers. And on that tropomyosin are the troponin. Very, very important when we deal with contraction about the role of troponin. So here and there at intervals you see troponin. I have indicated with black to stand out. In the model it was the white thermocol balls. It is attached to the tropomyosin and the active sites. Very, very important. So this is the real or pull out. Next, the myromyosin from the pull out. It comprises the head, short arm. This constitutes the heavy myromyosin, and the tail, which constitutes the light myromyosin. That way you see how are they organized. See, many of them are organized together and the heads are popping out at right angles to each other at intervals. See, this is the way it is organized. So this is the myromyosin organized into myosin. Now we have, I have already explained. Now the myosin and the actin, now how are they aligned? This is the way it is aligned. See, you have the actin or the thin filament with all the 4 proteins together, and then we have the myosin with the myromyosins polymerized to form the myosin. Very simple is the structure of the myosin. Then very carefully you see the way it is aligned. Look at the head. Head is correctly aligned in close proximity to the active sites. See here the tropomyosin is indicated with yellow color. I am just drawing with blue. This is the tropomyosin which I used in my model, the blue ribbon. So I am indicating here also the tropomyosin. And on the tropomyosin is what is bound our troponin, which is indicated here in green color. Here and there at regular intervals on the tropomyosin, we have the troponin. And look at the location of the troponin and the myosin head in very close proximity. The reason is that during contraction, this troponin is going to be removed and the active sites are going to be exposed. So if it is removed, I will make it like a cup. It is removed, vacant cup, vacant space, and into this vacant exposed site, active site, this is called the active site, into this exposed site the head is going to bind. See, all the head is going to bind. So this is going to play a very important role in contraction when we will be learning in the next video sliding filament theory. I hope you have understood the basic architecture of the skeletal muscle and the 2 proteins or the filaments and the proteins they comprise. Thank you.